Good afternoon, California. Thank you very much for having me. Andrew, thanks for the invitation. I apologize for not being able to attend in presence due to some personal circumstances. Greetings to Todd, greetings to Maddie. I know you personally. Would have loved to see you and chat about research and life. Unfortunately, it's what it is now. So, continuous water uptake information of plants, and this is kind of lessons learned from applying in situ water isotope methods for almost 10 years now. I'd like to acknowledge my co-authors who did most of the field work and also did quite some mentoring in the last decade. On the left you see an image of myself. I lead the research group Isodrones. We apply isotopes and drones and sometimes together also to understand the water cycle of different environments better. And I would like to direct you to our YouTube channel in particular, where you find a lot of videos from our group that might be interesting for one or two of you. In the background you see our first in situ campaign in Sweden, northern Sweden, where we studied water uptake of pines, lateral water uptake in that case with in situ methods. It looks kind of crazy. I hope it becomes clear during the presentation. Main motivation, you have had droughts in California recently and forest fires. We also had droughts in Europe. 2018 to 2020 were the most severe drought period in 250 years. And the result you can see in the left image, trees dying, complete landscapes are changing. Here the example of the Harz Mountains. It's a very touristy region where unfortunately they had planted almost exclusively spruce trees, and it looks horrible there right now. As legacy effects, not only the shallow rooters were suffering, but we also see this now in beech trees. We see the complex diseases, we see the ash dieback, we see severe consequences for oak trees. So across the spectra, trees are affected. And we need to find ways on improving that. And one step towards improving that is to understand where they actually get their water from and where are the water resources that they could eventually use and they are using. This review on the right side I really liked because it was investigating globally water stored in the rooting zones of vegetation, and what they found is that almost 40% come from water stores deeper than 2 meters depth. They also concluded that the water stored in the root zone is largely unknown, and that, as a matter of fact, because we are lacking methods to observe that, it's very difficult. And as a consequence, this is also not fully represented in models. Plasticity, not represented at all. And with these models, we do our predictions of decades, of centuries, but they are so uncertain because we don't have this root water uptake information. So the question here is, which water sources are plants using, and how can we investigate that or improve their investigation? I'm an isotope ecohydrologist, so my preferred method is water stable isotopes. And you see here on the left side, left image, a classical picture of the isotope distribution in a soil profile. You have more enriched water isotopes in the shallow surface because evaporation preferentially evaporates the lighter isotopes, and then at depth you have a more depleted or lighter isotopic composition. If we get now trees into the picture, you see a shallow rooter here using preferentially or almost exclusively this enriched water on the shallow surface, and you have a deep rooting tree which has also access to a deep reservoir. As a consequence, if we take a sample from the trees, you will find different isotopic compositions. The shallow rooting tree will have a heavier isotopic composition, and the deep rooting tree will have a lighter isotopic composition. If we have the information from a trunk of a tree and from a soil profile, we can determine how much water is coming from which depth by applying statistical methods or also simpler methods. What we often do is we increase the difference, or we increase the heterogeneity of the isotope depth profile in the soils by applying deuterium, often as irrigation, but sometimes also applied at depth, to become a much more clearer picture of water uptake. Traditionally, and Todd has applied this very often, we were collecting soil profiles, took a sample from the tree trunk, then brought this to the laboratory, it extracted the water, measured it in the isotope analyzer, and then we can apply mixing models, for example, or simpler methods to determine how much water is coming from which depth. In this example, 50% from 1 to 4 meters, about 40% from the really deep subsurface. This was an acacia, they are known for deep rooting. What we said is, this is very laborious, there are some methodological artifacts that have been discovered, and we need certainly new methods to increase the temporal resolution, because we cannot drill every day into this tree, but we know that trees are very plastic in terms of their water uptake depths. So how can we get a higher temporal resolution of this data? So what we did, and in particular my PhD student Kati, who defended in July with distinction, said we bring this isotope analyzer to the field instead of bringing all the samples to the laboratory. Sounds simple, it's not. And she has worked 5 years on this, and we are really happy with the outcomes. So here you see many trees connected to this isotope analyzer, and I will speak about this development for the rest of the time. The first time we tried this, we said we have to prove that this concept is working. And the idea that Mr. John Marshall had is to make a stem borehole. That's why the method is called borehole equilibration, which means drilling through the whole stem, make connections on both sides, be able to insert dry air from one side, and to capture the air coming out of the tree from the other side and send it to the isotope analyzer. What happens inside of the tree is that isotopic exchange happens with the dry air that comes in, and eventually it reaches isotopic equilibrium, and this is what we want to measure in the isotope analyzer, because from measuring water vapor we can infer the liquid water isotope composition under equilibrium conditions. We tested how fast the water vapor inflow can be in order to achieve these equilibrium conditions. So we made a mathematical model including these processes, and we found a maximum flow rate that we can use to say isotopic equilibrium happens if we use less flow than this maximum flow rate. And we tested this for 3 tree species. In particular, we exchanged their complete source water, so these were smaller trees, and we took them, put them in a pot with water that was provided air with a pump, and we then measured over 1 month how this isotopic composition is changing. The results of this you see here. I only show the data for the pine. I admit that was also the species where it worked best, but it worked for all of the 3 species. What you see here is the initial source water that the trees were using. And up here with the dotted line, the changed source water isotopic composition. And what you can see here, basically, in this case, we installed 2 stem boreholes, one lower and one higher on the tree's trunk. And you can see that over time, the isotope value approaches our changed source water, and it reaches it and stays constant there. Amazing that this worked. We were so stoked because that was a practical proof. We did the theoretical proof. So then we went to the field, and this study is from a tropical dry forest in Costa Rica. This is how a typical monitoring setup looks like that we're using. We always combine quantitative information, soil water content and subflow, with more qualitative information, the water isotope values in the soil, which we measure with gas permeable probes, and the xylem water isotopes, which we obtain with the stem borehole method. And you can apply this in different ways. You don't have to drill through the whole tree. You can also make a 10 centimeter hole and put plugs with an inlet and an outlet, or you put membranes. The development really has improved in the last years, and yeah, basically this is what we need, because with this we can make a mass balance, not only the isotopic balance, and we can really say how much water is taking up from which depth in the soil. As I said, this was in Costa Rica. We chose 2 species which were keeping their leaves in the dry season, but one is a deciduous, facultative deciduous, which is called caoba, or Swietenia macrophylla, and an evergreen tree species which is called tempisca, or Sideroxylon capiri. And we monitored them for about 3 months, and at the end of the dry season, before the rain started, we applied isotopic label with the yellow submarine here, about 400 per mil in deuterium, and we irrigated the whole area. We tried to do this homogeneously. You see here the results. We had 12 trees, 2 soil profiles, and on the left column you see the facultative deciduous, on the right column you see the isotope values of the evergreen, and up here with the stars is the isotopic labeling that we applied. You can see after the application immediately in the trunk, in the lateral root, and also in the supposedly taproot, the isotopic composition in this species increases by magnitudes. So clear uptake of the water that we provided, and in contrast to that, our evergreen species almost takes up nothing from the label. That was pretty cool to get such a difference, because we really did not know what we would measure at that time. But I think this is a clear image of water use, shallow water use versus no shallow water use. So there are no roots there, and the evergreen tree just does not take up this water. This is backed up by the subflow data. So the facultative deciduous takes up the subflow rate of the deciduous increases immediately after the irrigations. For the evergreen it stays constant, and it even decreases for this one tree. This tree actually exchanged its leaves during the monitoring period. This is all written down. This was pretty cool. It's not a proof of deep rooting, but it's a proof of how we can use these in situ methods to really monitor changes in water uptake depths and to prove which water they are using. We also applied this in a gigantic experiment in the biosphere too. There's a rainforest growing inside of this greenhouse, and this is published in Science together with the group of Christiane Werner. And there are also some other publications related to that. Here we applied a big amount of deuterium of about 1000 per mil via the drainage system of this biosphere, and through this we reached a homogeneous label at the surface of the biosphere, which is in 3 to 4 meters, and then we monitored what the trees do when an atmospheric drought is artificially introduced in this system. It was a great experiment. We learned a lot. We learned a lot how these trees behave, and unfortunately I don't have time to present this today, but with this I want to wrap up. I hope I got you a bit excited on in situ measurements of water isotopes. It's possible. It's still a lot of work, and it's work intensive, and it's difficult. You have to be there. It's not really unattended, but it has a really high potential if you apply these continuous measurements as complementary data sets in particular, and when you combine it with labeling. It has been shown that they improve the model performance significantly, in particular the root water uptake part, and there are also methodological things that don't apply to the in situ methods, which make them my method of choice. We have to investigate better legacy effects, which is true for destructive samples also. But I leave it there. Thank you very much. talk to you soon and hit me up for questions. Thank you from the team.